Hello everyone. This is about the biolab projects that we work on in the labs. About the transformations of E. coli cells with the HsRad52 mutant plasmids that we made in the lab. This is Susan Win, and alright. So the steps, we are trying to make a mutant that a cultures of bacteria that could express the HsRad52 mutant proteins inside of them. So we are trying to create one recombinant plasmid and put it inside of the host E. coli cells and transform the cells so that we could test it further. Alright, the first step is the DNA extraction. It is the plasmid DNA extraction. The plasmid is the circular piece of DNA that the bacteria could use and express the genes out of it. So at first we used a well-grown bacteria culture. We suspended those cultures and we lysed it with detergents and NaOH, which are gonna make the DNA's strands come apart. So after that, we reneutralized it with hydrogen chloride, and those DNA strands will come back together. However, the chromosomal DNA of the bacteria would be too large to actually come back together, and it will be lumped with other cells junk, the cell membranes and all those stuff. So after we centrifuge it, it would stay in the pellet. So we extract the supernatant and we run to the nucleospin columns and bind the DNA onto the membrane right there. And then we wash the membrane with a bunch of other buffers that will dissolve the proteins and the salts and the everything that is not DNA. So after that, we will supposedly only have the DNA right there, and then we will use the elution buffer so that and then centrifuge, and we will collect the pure plasmid DNA. Alright, from the result of this, we use a little samples of that purified extracted DNA and run it on the gel electrophoresis. We loaded those DNA with the loading die inside of this flow and ran the voltage to it, so the DNA will run down. And as you can see, this is lane 1 is PGBT9 HsRad52. This is the vector plasmids that vector of the plasmid that we want to make, and in the lane 2 is the place 744 is the insert part of what we part of the plasmid that we want to make. Alright, as you can see, this is the in the lane 1 there's not much DNA you could see, but possibly that I didn't load the actual DNA in because I didn't miss it hard enough, but clearly you could see there's like some kind of band in the lane 2, so I have a play plasmid for sure. Alright, moving on to the next step, which is cleavage of DNA with restriction enzymes. So what is it essentially is we want to cut the DNA, the desired DNA out from those two whole plasmid. So this one is what we have the mutated versions of human Rad52. So right here, the play I worked on is like play 744, but this is the general map of the play plasmids. So we will use the mutated versions right there, so we will cut it right here and there with the restriction sites of SAC1 and APA1 restriction enzyme site. And also from this vector plasmid that will hold the insert of the other, and we will also cut it with APA1 or SAC1. It will fit right in here. and we want to isolate the big piece here and the small piece right there. We use this plasmid because there's like a Gal4 binding sites that could test the functions of the Rad52 plasmid, and the ampicillin restriction sites, which will select the bacteria that only contain this our created plasmid inside in the presence of ampicillin, of course. Alright, moving on, what we did is we put in a APA1 and SAC1 with their optimal buffer and their optimal temperature in the water bed, and then we deactivated the enzymes. Alright, now supposedly we have the cut pieces of DNA inside of our epitope tubes. So let's see the result. This is the results of the PGPT9 Rad52, and as you can see, this is the uncut DNA. This is kind of spread out, it's not too yeah, and this one is the digest DNA. As you can see here, this is a smaller piece and this is a bigger piece, so we want the biggest piece because this is a vector version of it. So, and then the other one is place 744, which doesn't look good because we didn't load enough, or there's like not much times, does need more time to run because there's like all those pieces are bunched together. However, we want the smallest piece, so this is not gonna work, but fortunately the instructor kind of made another gel for us so that we could isolate and cut out the pieces that we want to isolate. Alright, next step is gel fragment purification. We use this freeze and squeeze spin column. Supposedly, if you cut out the gel piece that have the DNA in it and put it inside here, and then centrifuge, and you will have the purified DNA collected right there in the dolphin tube. Alright, so we try and say we get some liquid, but supposedly those are the DNA we want, but after we extracted and purified, we loaded on the gel and tried to see it, but we didn't see anything. It could be that we put in very little concentrations of the DNA inside or not, but we are moving on to the ligation part. The ligation is essentially we put the vector and the insert together and use the ligase to add this two up so that it could become a whole new plasmid. So we have two controls and two concentrations of the ligation mix. We put in a little bit more of the inserts inside of the ligation 1 and 2. Supposedly they will make the whole new plasmid that we desire. And this ligation 3 and ligation 4, I do it for the PGPT9, and I only put in vector DNA inside of these two tubes without any insert. So this is to test that the vector DNA, if it is come together by itself without the ligase, or by itself with the ligase without any inserts in it. So we didn't see the results at first. We use those ligations and try to put it inside of the bacteria, put it in the water bath to incubate and stuff like that. This is transformation. The transformation, you go in and go out as the Superman, right? So supposedly the bacteria will take up the DNA under the stressful condition, so we put them in a stressful condition so that they could take up our ligated plasmids inside. As they take up the ligation plasmid inside, they should have an ampicillin resistant gene, and when we plate the cells with the ampicillins and the food on the plate, they should survive. So for the result, this is the controls to see like if the cells are actually survive. It actually could survive, and the ampicillins are actually working right now. Like, let's see the bacteria, the host bacteria that didn't have any ampicillins on it, they could survive pretty well. And this one have ampicillin on it, they die. Poor guys. Alright, but this one have the plasmids of some kinds with the ampicillin resistant gene, so they could actually take up the gene and they survive, as you can see here. So, but unfortunately, our result is not look good. The ligation 3 and 4, they are not supposed to have a complete plasmid, so the bacteria should not be able to survive in the ampicillins, so they did not. Great. But for ligation 1 and 2, they are supposed to survive because they're supposed to take up our plasmid, but they did not. It might be that our plating strategy is really bad, and they die in the process of our plating. Or it might be that the freeze and squeeze kind of purification, as you can see, there's no DNA inside, so it might be that one is went wrong, or it might be really low concentrations and we cannot even make a complete legit plasmid. Anyhow, that's what we end up with, and we are trying to make this new beautiful DNA that could have those selection sites and binding domain that could test the proteins of it and put it in the cells to have a transformed cell, so that we could examine the products of those mutated versions of human HsRad52. However, we didn't get to do that, which is sad, but yeah, this is science and this is research, so what do you expect, right? Alright, thank you so much for watching, and that's pretty